All right. We're going to hear from Wayne Fuchs next.
Welcome back, everybody. First, I would like to thank Sharon Terry for inviting me to participate, and the entire staff at PXE International for executing this sensational virtual event. Today, I will briefly talk about research pertinent to patients with PXE concerned about vision loss. Before I start, there were several questions yesterday about loss of vision from retinal pigment epithelial atrophy. This is a patient of mine that was reported in the literature who was losing vision in both eyes. Here you see a color photo on the left.
We don't see any slides yet, Wayne.
No?
Not yet.
Let me get back somehow. Let's see, share screen.
There we go. It's starting now. We see a wonderful baby feeding another baby.
Do you see this now?
Yes, we do. Thanks.
Let's see the baby picture. All right, well, we already welcomed everybody. Let's see if we can, we'll stop that and we'll get on to this. So we already, now do you see the picture on the left?
Yep, we can see it.
Okay, so here, this is the patient I was describing with vision loss. And then the color photo on the left and corresponding red-free photo on the right, you can see the angioid streaks. And what I'm pointing out now is an area of lighter pigment through which you can see some choroidal vasculature. The reason you can see through the retina and visualize the choroidal vessels is because there's loss of the retinal pigment epithelium. You can see the very border of this well-circumscribed area of atrophy in both the color and red-free pictures, which is adjacent to the center of the macula in this patient, what we would refer to as juxtafoveal. The fluorescein angiogram brings out the margin of this atrophic area due to the loss of the pigment epithelium. You remember that the RPE and rods cones or receptors are a complex. You can easily visualize the choroidal vessels now filling as the fluorescein dye is not blocked by the pigment. And you can see the tapering edges of the angioid streaks. On the right, you may note in the later phase of the angiogram, some of the fluorescein has leaked out of the choroidal vessels and stained the sclera, or the white part of the eye. And you can see the choroidal vessels as a silhouette. Let's move on to a few projects that I have been involved with. Yesterday, I showed slides of the first patient I was involved with in 1984. He had angioid streaks with no skin manifestations and was one of a series of patients that we reported in the New England Journal of Medicine whose diagnosis was established with the biopsy of a scar. You can see the clumping and fragmentation of elastin fibers in the deep dermis in this histopathology slide on the right side of your screen, taken from a biopsy of his right forearm. When I say we reported, I was standing on the shoulders of clinician scientists who are considered giants in the field, co-authors of this paper, Drs. Mark Lebwall, Larry Yanuzzi, and Stanley Chang. But my interest in PXE was born with that patient who was both a challenge to diagnose and treat. I later became involved with the search for the genetic defect responsible for PXE. My role as a clinician in that project was to recruit affected individuals to bring family members for eye exams. I and many other ophthalmologists would examine the eyes of the extended family for ocular manifestations to determine who had evidence of PXE and who did not. Blood samples were drawn from the family and sent to the basic science researchers for analysis, and they discovered the gene on the short arm of chromosome 16 and are now working on a cure. I was later involved as a co-investigator in 2 treatment trials, neither of which have shown a clinically significant response, although there was a positive trend in the magnesium trial, as Yoni just mentioned. Magnesium is not currently recommended or approved by the FDA for treatment of PXE. To get a feeling of the level of interest in the real world about current research, I would guide you to a website, Trials.gov, which lists the ongoing research projects. If you search for angioid streaks, for example, you come up with a list of only 10 projects, 3 of which have been withdrawn and 1 of unknown status. As Sharon Terry mentioned yesterday, PXE is just one of thousands of rare diseases. This is why the basic science research being performed around the world, and especially at the PXE International supported Center of Excellence at Thomas Jefferson University, is so important. As you can see, most of the studies are evaluating the injection of a variety of drugs for treatment of choroidal neovascularization in patients with PXE and angioid streaks. These studies may be important not only in guiding our choice of drug, but may encourage insurance companies to cover the cost of these expensive medications, which are approved for patients with macular degeneration to be used for choroidal neovascularization secondary to angioid streaks. In contrast, there are 1,754 studies of age-related macular degeneration listed on the website. Most of these investigations are for therapeutics, such as pharmacologic solutions, devices, or relate to RPE transplantation, stem cell treatments, or gene therapy. Fortunately, for patients experiencing vision loss, the results of these investigations can be extrapolated to patients with PXE due to the similarities in the pathology of the 2 conditions. We will briefly discuss some of these future approaches to complement our current treatment strategies, which, as we spoke about yesterday, employ the use of intravitreal anti-VEGF injections. On the pharmacologic front, we can employ longer-lasting agents with better delivery systems. De Kogan demonstrated that with the use of cell-penetrating peptides, relevant concentrations of anti-VEGF drugs could be delivered to the back of the eye in mouse, rat, and pig eyes. Oral pyrophosphate, which has been spoken about this morning, a potent mineralization inhibitor, appears to be safe and well tolerated and may reduce ectopic mineralization. Alternate ways to increase circulating pyrophosphate, either by exercise, which releases pyrophosphate from erythrocytes, or tissue nonspecific alkaline phosphatase inhibitors, are potential treatments. Magnesium supplements need further study in a larger trial. Surgical approaches, especially with the advent of anti-VEGF agents preventing hemorrhagic complications and more patients reporting vision loss from pigment atrophy, include RPE transplantation. Human embryonic stem cells and induced pluripotential stem cells have become attractive sources for deriving RPE. They can both self-renew and differentiate into adult cell types. There has been much press coverage of the Moorfield Eye Hospital project using embryonic stem cell-derived RPE patches as a regenerative strategy. Of course, again, for age-related macular degeneration. A little plug for Mount Sinai and Dr. Bo Chen's group, who published this in Nature. They successfully reprogrammed Muller glia into rod photoreceptors and restored vision, not just in zebrafish, but in mice. Which brings us to a team of researchers led by Drs. Wito and Lee, who demonstrated that adenovirus-mediated ABCC6 gene therapy showed high levels of expression in the liver and restored plasma pyrophosphate levels, preventing ectopic mineralization in mice. Which leads us to another team of researchers across the river from Jefferson at the Shea Eye Institute, who have applied their laboratory experiments in mice to strategies in humans. What once was a dream has become reality, thanks to a dedicated team of researchers at University of Pennsylvania, the first gene therapy approved in the US to target a disease caused by mutations in a specific gene. Luxturna, a gene therapy medication for the treatment of Leber congenital amaurosis, was developed by Spark Therapeutics. It is the first in vivo gene therapy approved by the FDA in December of 2017. As Dr. Jean Bennett stated in her presentation at the Breslau Vision Science Symposium hosted by Lighthouse Guild in New York, it took 3 decades of hard work and 17.5 years from proof of concept to FDA approval. However, this milestone does create a path for future genetic treatments of inherited retinal disease. In a recent article in Retina, Dr. Hafler stated, as retinal gene therapies become available for patients with inherited retinal dystrophies, patients will increasingly benefit from genetic diagnoses. The goal of regenerative medicine are to either prevent atrophy or genetically programmed degeneration, or restore what has been depleted by atrophy, to use trophic factors to regenerate what is still capable of growing, and use stem cells to repopulate tissues. Gene therapy may correct the problem and prevent disease or further degeneration of tissues. New techniques such as CRISPR, clustered regularly into spaced short palindromic repeats, hold great promise for the future. This technique allows for the removal of abnormal DNA and replacing it with normal material, editing a specific bit of DNA inside the cell. You heard about this technique this morning from Olivia. This is currently in clinical trials. In addition to thanking everyone for their attention, I would like to express my gratitude to PXE International for inviting me and to my patients for their confidence over the past 35 years. I would especially like to thank this beautiful grandmother, Patty, the love of my life, for her patience, love, and support over the past 4 decades. Thank you.
Thank you, Wayne. And we also really appreciate Patty. Those of us who have been to your office have met her and are very appreciative for her support of you and for all the people with PXE. She's a wonderfully warm and compassionate woman. And you have beautiful grandchildren. I love grandchildren in these days, so fabulous.
Thank you.
We have a couple questions. Any studies being done to replace lost rods and cones?
Well, I mentioned the work in the Mount Sinai labs to replace where we did replace photoreceptors with glial tissue. Again, in mammals, certainly far from human trials, as you've heard from everybody this morning. Research takes a long time. So I would say, I tell my patients jokingly, we're not 3 weeks away, but it's probably less than a decade away.
Okay, great. Any other questions? Question about flashers and floaters and what they might mean for people with PXE.
Well, anybody with flashing lights and a new onset of floaters should have that checked. It's not specific for PXE, but it could represent the vitreous jelly condensing and collapsing and moving forward. And that happens to pretty much everybody eventually. But when it happens, that's when the patient is at risk for a torn retina. So we like to examine patients for that. If we find a torn retina, which is unlikely, maybe less than 10% of patients with symptomatic vitreous detachment. We call that a vitreous detachment, but if we find a torn retina, we would recommend treatment, usually with a laser. The idea is to prevent a detached retina. Laser treatment for a torn retina takes a few minutes in the office, and it's a lot easier than repairing a retinal detachment in the operating room, which is a more complicated procedure, more risks involved.
Great. Could you talk about your comment about FDA and magnesium?
Well, we discussed yesterday the magnesium trial. Yoni mentioned that it only had 40 patients. It did not show what we consider clinically significant improvement, but there was a trend towards reduced calcification in patients. And I've spoken with Mark Lebel about this, and he feels strongly that it would be beneficial, but certainly not proven to be statistically significant. It would require a larger trial to demonstrate that. So we currently do not recommend. I heard that the scientists have agreed not to make that recommendation for supplementation. Yoni mentioned that we get enough magnesium for the daily requirements in our normal diet.
Right. And the FDA does make minimum daily requirement recommendations, and magnesium is part of that. And we just say that that's a good thing to follow. You cut out when you were answering the rods and cones question. I think not for everyone, but for the person who asked the question. So if you wouldn't mind just repeating the nugget of that, whether or not they can be replaced.
Well, that was the publication I alluded to, and the reference is there to read. Bo Chen at Mount Sinai did do experiments where they converted glial cells, which are retinal cells, into photoreceptor cells in mice. I mentioned that it is an important step from zebrafish to mice, but we're not quite ready for human trials yet.
Great. Any risk of for cataract surgery in PXE people?
We talked about this yesterday, so we recommend if someone has had treatment with injections for active neovascularization, not everybody universally, but I would recommend to patients that they receive an injection 1 to 2 weeks before cataract surgery. Because even though cataract surgery is done through a small incision and doesn't even require sutures, the incision is so small, there is some healing and inflammation afterwards. And inflammation has been implicated in neovascularization. So to prevent a resurgence of active neovascularization in patients who have received injections, we recommend an injection prior to surgery. Cataract surgery is safe. Patients should be warned about having realistic expectations if they have macular damage from previous scarring. if they've had a previous bleed, previous atrophy, and there's damage to the macula. You can remove the cataract safely and get rid of that component of the decreased vision. But the chain is as weak as the weakest link. There will be some limitation from of the benefit from cataract surgery.
Great, thank you. Any other questions? Okay.
Yes, Sharon, I have a simple one.
Sure.
Wayne, this refers back to yesterday, if you don't mind. You said that Avastin usually costs around 50, Lucentis around 2,000. But if you said it, I missed it. How much does Eylea cost per injection?
Right, I had emailed Sharon this morning about that. So the three medications approved by the FDA are in the same ballpark. So when Lucentis was first approved, made by Genentech, it was $1,930. Regeneron came up with Eylea maybe 9 or 10 years ago, and it was priced at $1,850. And so they're smart. They were trying to break into the market, and they made it a little less expensive, even though it lasted a little longer. And Novartis did the same thing with Beovu, again in the $1,700 to $1,800 range.
And it's quite interesting that there's lots of arguments within FDA and within the patient groups that I work with, because we spend time discussing this with FDA and the drug companies, that the drug companies claim they're going for value-based pricing. They're essentially going for what will the insurance market bear, that they don't have to fight the insurance companies for. Europe usually negotiates much lower prices than the US does, because the US is very supportive of pharmaceutical companies and this administration. And so these higher prices are being allowed, and in fact, in some cases also encouraged. We are very concerned that as rare diseases get drugs, and now 237 rare diseases have drugs, those are between $100,000 and $1 million a year, that in fact will actually break the GDP of the US if we find treatments for even twice as many rare diseases. Lots of people, especially in the US, say, well, it doesn't cost me anything, it only costs my insurance company. But we all know that there is an interconnectivity between these things. And as those prices go up, the insurance companies are not going to be able to afford to cover other things that are more common. And we're going to be in a worse crisis than we already are in terms of healthcare. And then in Europe, there's much better control by the EMA, which is the European Medicines Agency, and the negotiations that the European Commission has done around these prices, and they get it for remarkably less than we do in the US.
I would add that the drug I mentioned, Luxturna, for Leber's amaurosis, costs $850,000. So it is one injection, it's not repeated, but insurance companies have arranged payment plans when treating patients with that. So it is a problem.
And it's quite remarkable too, in these conversations, often the drug companies will say, people should just remortgage their house because they'll be saving their vision or saving their child or saving their mother. And at some point, these kinds of costs really are not so tenable. I had a conversation with the company that created the cystic fibrosis drug, Kalydeco. And when it was under development, the researcher said to me, if we get $80,000 a year, we'll be getting 10 times what we paid to make this drug. And they actually put the drug on the market for $690,000 when it did come out, because they knew they could make a killing on it, since it was a very good drug for a certain proportion of cystic fibrosis patients. So we need to see radical change, and it's super hard to get it going. I keep talking to Congress about it, but we'll see as we go. Good question. Thanks, Pat. All right. So we are at the point. Thanks very much, Wayne, and thanks to speakers so far.